Hello and welcome to Unacademy, a one-stop destination for all English medium civil service aspirants. Welcome to Science and Technology Simplified. In today's session, we'll discuss genetic engineering and its applications. You can find the annotated PDF of this session in my Telegram channel, the link for which is provided here. Before I start the session, a very important announcement. You can now get all our GS and CSAT courses for just 17,999 plus free access to our exclusive UPSC mains course, the Nischay 2026. This is a limited period offer, so do enroll before it is late. Don't forget to use the code IAS English. And do participate in our mega lottery as a part of Dil Se Diwali, the Unacademy Utsav 2025. Let us now start the session about the genetic engineering. What is genetic engineering? Genetic engineering is manipulating an organism's genes or modifying an organism's genes is called as genetic engineering. The genetic engineering process involves adding the genes which are necessary, deleting the genes which are not needed, and modifying certain genes to give new characteristics to an organism. Genetic engineering will create new types of organisms that do not exist in the nature. In brief, that is about genetic engineering. And genetic engineering is not a single technique. This includes several techniques. Let us see some of those techniques. The first one being recombinant DNA technology. The name itself is self-explanatory. Recombinant DNA technology means making new combinations of DNA. Therefore, it is called recombinant DNA technology. In this technology, we make new combinations of DNA. Therefore, this technology is called recombinant DNA technology. Then what do you mean by new combinations of DNA? For example, a bacterial gene in a plant DNA or a bacterial gene in an animal DNA. This kind of combinations we create in this technology. Therefore, it is called recombinant DNA technology. And gene cloning is also a part of the recombinant DNA technology. In gene cloning also, we will create new combinations of DNA. Let us try to understand what is recombinant DNA technology and gene cloning with the help of this diagram. If you observe, this is a human cell. Let us assume this is a healthy human cell. Inside that, I have indicated a thread-like structure, that is the DNA of that cell. We all know that genes are present on the DNA. In the human DNA, we have a total of 30,000 genes. But as I cannot show all the 30,000 genes here, I have shown one gene, insulin gene. Like this, we have several genes on the DNA. But right now, our discussion is about the insulin gene. Then, now, in the first step of this procedure, what we do is, we cut the insulin gene. Step 1, what we do is, cut the insulin gene. Step 2. For the step 2, look at this cell. This is a bacterial cell. In that, there is a thread-like structure. This is nothing but the bacterial chromosome. Bacterial chromosome. In addition to bacterial chromosome, the bacteria also contain another small piece of DNA called as plasmid. The second step is cut the plasmid. Cut the plasmid. Let us imagine my hand, this is the plasmid. What we have done? Cut the plasmid like this. Then if you remember, in the first step, we have already cut the insulin gene. Let us now imagine this is the insulin gene. Step 3 is combine both of them. Simple. Cut the insulin gene, cut this gene and then cut this plasmid, cut the plasmid, cut the insulin gene, attach both of them. So, third point is attach the gene plus plasmid. This is human gene. What gene we have attached? Human gene. In which plasmid we have attached? Plasmid is the bacterial DNA. Plasmid is a bacterial DNA. Means it is a new combination of DNA. Therefore, we call this combination as recombinant DNA. Recombinant DNA, a new combination of DNA. If you observe, this green color structure that I have drawn here is nothing but the bacterial plasmid. In that bacterial plasmid, we kept this insulin gene. Insulin gene is kept in the bacterial plasmid. Bacterial plasmid plus human insulin gene together is called recombinant DNA. Then what is the step 4 here? The step 4 is insert that recombinant DNA into the bacterial cell. Insert the recombinant DNA into the bacterial cell. This will form recombinant bacteria. Look at here. What we have done here. Insert this DNA. Insert that recombinant DNA into the bacterial cell. If you observe, this recombinant DNA is now, inside the bacterial cell. This recombinant DNA is now inside the bacterial cell. The bacteria containing the recombinant DNA is called recombinant bacteria. This is the recombinant bacteria with human insulin gene. A bacterium with human insulin gene. So we call it as recombinant bacteria. Okay. Then fifth step is let that bacterial cell, let the bacteria divide. Means it will undergo cell division. If one cell divide, it will form two cells. Then the 2 will become 4, 8, 16, 32, 64, 120, 1 million, 2 million, 4 million. Like that, the number of the bacterial cells increase. Then next step, sixth step is the bacteria will copy the human insulin gene. The bacteria copies human insulin gene. Why the bacteria copies human insulin gene? Because we kept this human insulin gene inside the bacterial cell. Now the innocent bacteria thinks that it is a part of its own DNA and will copy this every time it divides. So every time the bacteria doubles, our insulin gene also doubles. So we can call this particular procedure as, gene cloning. Gene cloning. Why this is called gene cloning? The word clone means copy. The word clone means copy. What are we cloning here? We are cloning. We are copying the gene. Therefore, this procedure is called as gene cloning. Which gene we have cloned? Human insulin gene. Who cloned it? The bacterial cell. Means we used the bacterial cell as a Xerox machine to copy our gene. Okay. And then in the next step, what happens? Seventh step. The bacteria will also produce the human insulin gene. The bacteria will produce the human insulin, not the gene, sorry, human insulin hormone. Human insulin hormone will be produced by the bacteria. It means the bacteria will do two things for us. It will copy the human insulin gene and then it will produce the human insulin hormone. This hormone can be used as an injection. This can be used as a drug for the Diabetics. Diabetics take insulin injection. That insulin injection is produced by this procedure. I repeat, cut the insulin gene. Okay, cut this insulin gene. Okay, then cut the plasmid. Then third step, attach both of them. This is called recombinant DNA. This is our recombinant DNA. Then transfer that recombinant DNA into a cell, bacterial cell, that makes it recombinant bacteria. This is recombinant bacteria because it has recombinant DNA inside. Then next step, let the bacteria multiply. Let the bacteria multiply. Along with the bacteria, our gene also multiplies. Therefore, we call this procedure as gene cloning because our gene is getting cloned. It means our gene is getting copied. And then the bacteria will also produce human insulin hormone, which can be used as a drug for the diabetic patients. This is gene cloning because we are copying the gene. And this can also be called as recombinant DNA technology because in this technology, we made a new combination of DNA by placing the human gene into the bacterial DNA. So the names recombinant DNA technology, gene cloning, both the names that I have used here are applicable for this particular technique. Gene cloning because it is copying the gene. Recombinant DNA technology because it is making a new combination of DNA in this technology. And now comes the question, is the recombinant DNA technology only for insulin hormone production? No, we can also use it to produce growth hormone, growth hormones to cure dwarfism, growth hormones to cure dwarfism. We can also produce the clotting factors, blood clotting factors, blood clotting factors to cure hemophilia, blood clotting factors to cure hemophilia. Hemophilia is a bleeding disorder where the people's blood will not clot. Blood clotting means what? If I get injured, I will bleed for 2 minutes. Then after that, the blood there will solidify and I will stop bleeding. This is called blood clotting. This occurs in us, but in hemophilia patients, it will not occur. Why it will not occur? Because a clotting factor is missing in them. So we can inject blood clotting factors. Or say for example, dwarfs. Why they are a dwarf? Because they don't have sufficient amount of growth hormone. So what we can do is, instead of the insulin gene, transfer growth hormone gene. Now you will form a recombinant DNA with growth hormone gene. And we have inserted the growth hormone gene in the bacteria. Now instead of this insulin hormone, I told you bacteria produce insulin hormone. In the place of that, now the bacteria will produce growth hormone. If you keep insulin gene, it will produce insulin hormone. If you insert growth hormone gene, then it will produce growth hormone. If you insert blood clotting factor gene, I mean in this place, instead of this insulin gene, if we use, instead of this insulin gene, if we use blood clotting factor gene, then this bacteria will contain the blood clotting factor gene So such bacteria will produce blood clotting factors. Like that, we can also produce vaccines. Such vaccines are called as recombinant vaccines. Recombinant vaccines, we can also call them as subunit vaccines. Subunit vaccines. How to produce these vaccines? Let us suppose this is the coronavirus. Imagine this is the coronavirus. Okay, and this is its spike protein. Spike protein. And this is its genetic material. genetic material. Now, on that, let us suppose there is one gene. Okay, this is the gene that produce the spike protein. The gene that produce this protein. Okay, and the important point is genes produce proteins. So don't get confused. This is the spike protein, and this red color one is the gene that produces spike protein. Now what we do? Cut the spike protein gene. Is it clear? Cut the spike protein gene. Step 2. Insert this in the bacterial plasmid. Then give it back to bacteria. Then the bacteria will produce, same like this, just like here. Okay, now just imagine if this bacteria contain the spike protein gene. Just imagine like that. If this is the spike protein gene of coronavirus, then what happens? Such a bacteria will now produce, the viral spike protein, and this spike protein can be used as a subunit vaccine. This is called a subunit vaccine because I am not injecting the total coronavirus into my body. I am injecting only this spike protein into my body. So this is a subunit, not the whole structure. Therefore, we call it a subunit vaccine. And we also call this as recombinant vaccine because this is made using recombinant DNA technology. What is the recombinant DNA that we made here? Imagine this is the bacterial plasmid, and in that I kept the viral spike protein gene. So what is recombinant DNA? A combination of two DNAs. Here I made a combination of a viral DNA and the bacterial plasmid. Bacterial plasmid is nothing but the bacterial DNA. So this is nothing but a recombinant DNA. This combination is nothing but a recombinant DNA. This recombinant DNA is kept in the bacteria. Then the bacteria will produce the coronaviral spike protein. Bacteria produces coronaviral spike protein. Extract it. That spike protein can be used as a vaccination to prevent the coronaviral infection or COVID-19. Okay. Names of such vaccines are Corbevax, Covovax are the examples of such vaccines. Examples of recombinant vaccines or subunit vaccines. Examples of recombinant vaccines or subunit vaccines. With that we understood what is recombinant DNA technology and what is gene cloning. Now the third technology under genetic engineering is gene therapy. If you treat any disease using chemicals, we call it as chemotherapy. If you treat any disease using radiation, we call it as radiotherapy. But if you treat any disease using genes, we call it as gene therapy. Okay, now let us see how the gene therapy is made. Imagine this is this one, this complete structure. This is the double-stranded DNA. This is one strand of the DNA. This is another strand of the DNA, and together this is a double-stranded DNA. We all know that genes are present on the DNA. Let us assume they are 1, 2, 3. What are they? This one, this part, this part, this part. What are they? I have shown 3 genes on the DNA. I have shown 3 genes on the DNA. Okay. Then let us suppose this one, the first gene, if you observe here, this gene is a mutated gene. What kind of a gene is that? That is a mutated gene. Okay, mutation means what? The word mutation means change in the gene sequence. Change in the gene sequence. In general, in our DNA, adenine will always pair with thymine and guanine will always pair with cytosine. So, adenine pairing with thymine correct, thymine with adenine correct, adenine with thymine correct according to this rule. Guanine with cytosine correct, but guanine should pair with what? Cytosine, isn't it? The guanine should pair with cytosine. It means the correct letter here is supposed to be cytosine. You should have cytosine here, but you have thymine. Means this is a mistake. And such mistakes are called as mutations. Such mistake in the gene sequence, we can compare it with a spelling mistake. It is supposed to be C, but you have T. That is like a mutation. For example, if this is a important gene, for example, if this is hemoglobin gene, and if that is mutated, then we will get diseases like sickle cell anemia, thalassemia, etc. We will get such diseases because of a mutation in the hemoglobin gene. So, let us imagine that this is a mutated hemoglobin gene. Okay, what is that? Mutated hemoglobin gene. Because of that mutated hemoglobin gene, we get diseases like sickle cell anemia, thalassemia, etc. They are the hemoglobin related defects. So, now what we have to do? Here the solution is not, in the gene cloning what we have done? We have given insulin injection, is it clear? Insulin hormone the bacteria produced and we have given insulin injection. But that is only a temporary solution. You have to give that injection every day. Is it clear? But if I want a permanent solution, I want to solve the problem at the root cause level, then what we have to do is gene therapy. It means don't give the superficial treatment like injecting whatever components are missing. Correct the mutation in the gene so that that human being himself will produce the product which is required, That concept is called as gene therapy. So in the gene therapy what we have to do? Okay, simple. Step 1, delete the mutated gene, right? Then step 2, add the correct gene. That's all. Delete the mutated gene. This gene, mutated gene, we have to remove and add the correct gene. What is the mutated gene? AATCT. Okay, that is the mutated gene. Why that is mutated gene? Because this is the mistake. I deleted that. Then what should I add? Correct gene. What is the correct gene here? AATCC. What should be the correct letter here? G will pair with C. You have G here. So the other strand here should have C. So what is gene therapy? Just delete the mutated gene, wrong gene, and add the correct gene. Wrong gene is this AATCT. Correct gene is this AATCC. Okay. Who will cut this mutated gene? Okay. Cas9 will cut the mutated gene. Cas9. Okay. This one. In the CRISPR Cas9, this Cas9 is a nuclease. Cas9 is a, what is Cas9? Cas9 is a nuclease. In the nuclease, ase means cut. Nuclei means nucleic acid. Which nucleic acid? We have two types of nucleic acids, DNA, RNA. It will cut the DNA. So Cas9 is an enzyme that can cut the DNA. Cas9 is an enzyme that cuts the DNA. Means Cas9 is like a scissor. Cas9 is like a scissor. So Cas9 cut the DNA, but Cas9 do not know where to cut. We have to cut this first gene, right? So by mistake if the Cas9 cuts the second gene, or by mistake if the Cas9 cuts the third gene, we don't want that. Okay, but Cas9 do not know where to cut. So we have another structure called CRISPR. What is CRISPR? CRISPR is a guider RNA. It's a guider RNA. What do you mean by guider RNA? Basically, it is an RNA. See, look at here. This is CRISPR. What is CRISPR? This one. UUAGA. This sequence is called as CRISPR. What is the CRISPR? It is a piece of RNA. CRISPR is a piece of RNA. Okay, basically it is an RNA. What is its duty? It will guide the Cas9. It will guide the Cas9 about where to cut. It will guide the Cas9 about where to cut. So CRISPR will guide. See, if you observe, where is the CRISPR bound? CRISPR bound to the first gene. So the Cas9 will understand, okay, I have to cut the first gene. This CRISPR is not bound to the second gene. So Cas9 will not cut the second gene. So CRISPR and Cas9 are like wife and husband. They are like a couple. They both are together. Wherever the CRISPR goes, there the Cas9 goes. CRISPR will show the Cas9 where to cut. It will show the mutated gene and Cas9 will cut the mutated gene. Now the mutated gene is deleted. We will add the correct gene. It means now they can produce healthy hemoglobin or healthy insulin. So like that we can treat the genetic diseases by modifying the genes. Because you are modifying the genes, we can also call it as genetic engineering. What is genetic engineering? Manipulating the genes, modifying the genes. That includes adding the genes, deleting the genes or modifying the genes. Here we have deleted a gene, we have added a gene. By that we have modified this organism, this human being, so that we made him again healthily produce the protein which he was unable to produce previously. This is called gene therapy. And if we do the gene therapy in the somatic cells of our body, somatic cells means what? The word soma means body. Therefore, somatic cells means body cells. Somatic cells means the normal body cells. Okay, means except the reproductive cells, all the cells except the reproductive cells. It means, for example, egg cell, sperm cell, zygote, these are the reproductive cells. Leave them. Remaining all normal body cells like my skin cell, blood cell, bone cells, liver cells, kidney cells, pancreatic cells, all of them are called as somatic cells. Okay, I told you the procedure of gene therapy. If we do this gene therapy in the somatic cells, we call it as somatic gene therapy. Same procedure. What procedure? Delete the mutated gene, add the correct gene using CRISPR-Cas9. If you do that in the embryonic cells, okay, then we call it as embryonic gene therapy. By the way, what is an embryo? During the childbirth, the egg cell of the mother will combine with the sperm cell of the father. They both will fuse like this to form a single cell called as zygote. Then the zygote will undergo cell division. It means it will become two cells. The two cells will again divide and that will form 4 cells. The 4 will again divide, that will form 8 cells. This is called as embryo. This is called as embryo. Okay, embryo is nothing but a group of cells. It's a group of cells. And from this embryo itself, okay, from this embryo itself, all cells of our body, all cells, all organs and all tissues of our body are formed from the embryo. So, if you delete the mutated gene and add the correct gene in the embryonic cells, then what happens? The correct gene will be reflected, that correction, that gene editing will be reflected in every cell of our body. Every cell will get the correct gene. So therefore, embryonic gene therapy is practically easier because we have to modify or edit lesser number of the cells. For example, you have to do this correction. What correction? Remove the mutated gene and add the correct gene. This gene editing, we can also call this process as what? Gene editing. Gene editing is a part of genetic engineering. It's a part of genetic engineering. Is it clear? So if this gene editing is done in the embryonic cells, you have to do it only 8 times. But if you do it in my body cells, you have to do this trillion times. The procedure is the same. If I do this procedure in the somatic cells, I have to do it trillions of times. But if I do it in the embryonic cells, I will have to do it only 4 times or 8 times. So therefore, somatic gene therapy is practically very difficult because of large number of somatic cells to be edited. I have to do this editing in trillions of somatic cells, whereas this is practically easier. Embryonic gene therapy is easier because you have to do that editing only in 8 cells. Is it clear? But here the drawback is embryonic gene therapy is not legally permitted because it can pave way to designer baby. Then what do you mean by designer baby? That is another application of genetic engineering. In the designer baby, we can have a baby with a list of all the expected features. Say I want a baby with fair skin, curly hair, blue eyes, chubby cheeks, very tall, very healthy, super intelligent. This kind of modifications I can do using the same CRISPR-Cas9 technology. But is it moral or not? Designer babies are not moral, because if we permit that, first of all, rich becomes more beautiful, more intelligent, more healthy, more stronger, more capable. So the rich becomes richer, poor becomes poorer. The advanced countries will become more advanced and the poor countries will not become advanced, because like their population cannot afford for the designer babies. In the rich countries, the population can afford for the designer babies. The population can become more intelligent, more capable. So this will create human inequality. We have already several types of inequality. This will add to another source of the inequality which we don't want. Is it clear? So that is the reason why embryonic gene therapy is not legally permitted. Somatic gene therapy is legally permitted but practically difficult. Let us just summarize. CRISPR-Cas9 technology is a technology for gene editing. Gene editing means we correct the mutations. There is a mutated gene here. We will delete this mutated gene. In that place, we add this correct gene. Who will cut this mutated gene? Cas9, because it is a nuclease enzyme, a DNA cutting enzyme, an enzyme that can cut the DNA like a scissor. But this does not know where to cut. What shows it where to cut? See, there is an RNA piece, a piece of RNA called CRISPR. The CRISPR will bind to whatever gene we have to cut. Then the Cas9 will cut the gene to which the CRISPR is added. Then a gap is created. In the gap we add this correct gene. This is called gene editing or gene therapy. If I do this gene therapy in somatic cells of my body, that is called somatic gene therapy. The disadvantage here is large number of the somatic cells to be edited. I have to repeat this procedure trillions of times. But the advantage is there are no legal implications. This is legal, we can perform that. Okay, and embryonic gene therapy is, procedure is the same, delete the mutated gene, add the correct gene, but using CRISPR-Cas9. If I do this technology in the embryonic cells of the human body, then that is called as embryonic gene therapy. Embryonic gene therapy advantage is it is easy because you don't have trillions of cells in the embryo. In 8 cells if you do this correction, that correction, the correct gene will be reflected in every cell of that baby who is going to take birth. Okay, therefore it is practically easier. I don't mean to say that it is a very easy technique, but it is easier when compared to somatic gene therapy. And the disadvantage here is it is not legally permitted. Why it is not legally permitted? It is good to prevent the genetic diseases before even the baby is born. It is quite good to do that. Then why it is not permitted? It can lead to designer babies. What are the designer babies? Designer babies are like a baby with all the expected features, like healthy, strong, beautiful, like that. And it will add to another source of human discrimination. So therefore, it is not supposed to be permitted. So therefore, though the embryonic gene therapy is very good, though we can do that, but because it can be misused to make the designer babies, therefore there is no legal permission for embryonic gene therapy. Not only embryonic gene therapy, we don't have legal permission for dealing with the or modifying the human embryos for any purpose. For any practical purposes, for any medical purposes, we do not have a legal permission to modify or edit or deal with the human embryos, because it can also lead to the death of the embryos, and also the designer babies can lead to some unwanted modifications. The greedy scientists can try to create some unwanted modifications in the human beings which can even backfire. For example, if you have the dark skin complexion and you may feel like I will make a designer baby so that my baby will have fair skin, but we have to remember that the dark skin is protecting us from the skin cancers. So it means we have certain features in our body because they have certain role, and if you modify them, what happens? Unwanted consequences can result. So that is the reason why designer baby should not be used. Therefore, because gene therapy can lead to designer babies, even embryonic gene therapy should not be permitted, but somatic gene therapy can be permitted. What is the applications of gene therapy? What are the applications of gene therapy? We can use this in cancer treatment. We can use it in the treatments like diabetes. We can also use it in the treatments like sickle cell anemia, thalassemia, etc. These are the applications of somatic gene therapy. So like any other technique, genetic engineering is also having its own pros and cons. So we cannot say any technique is good or bad by itself. It depends on the way we use it. That is all about genetic engineering and its applications in the human beings. That's all for today's session. Thank you so much.